Dry heat, usually what's happening is it's causing oxidation. So you're getting changes to the DNA, changes to the protein. It will also cause denaturation, right, because it's heat. So as an example, this is how we typically, right, in the lab, you hold your loop. I'm not teaching the lab, but I do know that's what you do. You hold the loop in the flame, right, to incinerate any microbes that would be on there. This is what's commonly done in clinical settings as well. Incineration of biohazardous waste. Oftentimes things aren't autoclaved, they are incinerated. Okay. In medical facilities, so gauze, patient samples, right, they can be incinerated. But it's the idea of using hot air. Now, you might be like, oh, so a hospital is just burning their waste, why won't they autoclave it? Well, autoclaving, especially depending on how much waste they have, you can probably get a bigger incinerator than you can autoclave. But an equivalent treatment, 170 degrees for at least 2 hours, would be equivalent to autoclaving for 15 minutes. So an autoclave is more efficient because you have that moist heat under pressure, but that dry heat could work as well. So incineration of biohazardous waste is a valid way to sterilize it. So that's using heat is one of the physical ways that we can control microbial growth, moist or dry. We can also control microbial growth using filtration. So there are going to be a lot of situations where something, a liquid, needs to be sterilized, but if you were to put it in the autoclave or try to boil it or something like that, you would actually destroy the components themselves. Antibiotics, vaccines, other medications, different kinds of growth media, depending on the types of bacteria that you're trying to grow, can be heat sensitive. But obviously, if you're going to have some sort of medicine injected into your body, you probably want it to be sterile. So one way that that can be done is through filtration. There are varying pore sizes. Typically, if we're making like a growth media in the lab, we really just want to be able to exclude bacteria. So we'll use a filter that has small enough pores to let the liquid through, but capture bacteria on the surface. If you were trying to eliminate any viruses, you have to use a much smaller pore. But for a research lab setting, or even for a clinical lab, if you're trying to like pour plates or something, viral contamination is not going to be a huge issue. You want to keep all the bacteria out. And for a long time, people used some of these like larger filters, these 0.45 microns. But it turns out there are bacteria that can pass through them. Things like Campylobacter, Helicobacter are very small. So you have to actually use a smaller filter pore if you're trying to filter sterilize. Okay, so again, that's the idea. It's also very difficult to autoclave air, right? The air that we breathe or the air that you might breathe in the hospital setting. So HEPA filtration can be used for that, to trap, as much as possible and remove it from gases and air. That's why masking, obviously I'm imagining most of us don't buy like HEPA masks, but if you buy a decent quality mask, it's not that that was the argument, oh, well, COVID's so small, it can pass through a mask. And I'm like, well, yeah, technically, but COVID's coming out in like your boogers and your saliva. The drops of that don't come out through the mask. So the viral particles are trapped within these droplets that don't get through the mask. And your bacteria don't get through the mask. So the masks are actually really effective at stopping those things. But yeah, if I had pure COVID viral particles that I was blowing through, they would travel through any one of the standard masks. But because they're associated with droplets, they'll stay in. So that's why masks are really beneficial at helping to prevent the spread of respiratory disease. And why on some job sites you have to wear a mask, or at least you should be. If you work outdoors, any sort of like field work, you should be wearing a mask because the fungi in the soil can make you very sick. So again, we would do filtration when we can't use heat. So when we have heat-sensitive materials. And the standard growth media that you guys will use in microbiology, even if some of you end up taking medical microbiology, most of that growth media is totally fine in an autoclave. But as you start to get media that requires various supplements, you might be able to autoclave to a point, and then you have to add filter sterilized supplements to it because you can't autoclave some of those supplements. So it can start to get a little bit more tedious, but because we have filtration, we're able to do that. And kind of on the opposite end of using high temperatures to control microbial growth, we can use low temperatures, but typically we are not talking about killing microorganisms. And I think we all know this. If you put food in the refrigerator, it will eventually go bad. If you put food in the freezer, it may not go bad in the freezer, but if you pull it out again, it will, even if you don't open it. Because most of the time, the temperatures that we can achieve using our commercial and household products is not low enough to kill microbes. The refrigerator, definitely not. The refrigerator can slow microbes. It can stop some. The freezer will stop almost everything, but not necessarily kill your standard household freezer. We have more powerful freezers here that would, but right, that's not the kind. You don't have a -80 in your house. There is an exception. Freezing meat can kill the larvae of a particular parasite. But there are some bacteria that do very, very well in cold temperatures that still cause human disease. I think earlier this semester I talked a little bit about listeria and how it has a really wide growth range. It can grow in the refrigerator. Like, it doesn't slow down, it grows between 1 and 45 degrees. There was an outbreak of listeriosis, foodborne illness caused by listeria in the summer of 2015 that killed people in a hospital setting because it was contaminating ice cream. So, I still eat ice cream, but in the processing facility where they made that ice cream, it was contaminated with listeria. So the ice cream was contaminated with listeria, and obviously you freeze ice cream. And this is ice cream it's a specific brand that's made in Texas and sold in like Texas and Oklahoma. So it's hot in those places, so you have to keep it frozen. And people died because the freezing doesn't kill the bacteria. So don't think of it as something that kills your bacteria. Okay, so we can talk about refrigeration, and that's typically what we use if we want to slow microbial growth of food products. Deep freezing, and that's again not like your chest freezer that you have at home. We have freezers here that again go down to like -80. That will kill most microorganisms. But that's so cold that if you touch it, like, it will start to kill your skin cells. It's very, very cold. It's counterintuitive, but it will burn you. It's like dry ice, right? It will burn you because it's so cold. And then lyophilizing is freeze drying. So they dry it out and freeze it. And that can kill them too. Or it can preserve food for a really long time. I know when I was a kid, they always made a big deal. You could get like the freeze-dried ice cream like the astronauts get, because the space program was really popular when I was a kid. So tastes disgusting. I'm like, oh, these poor astronauts have to eat this nasty ice cream, but it preserved it and they didn't get sick because you can't have food poisoning in space. I mean, you can. That would be very unpleasant. Zero gravity, no thanks. Okay, so, you know, just don't think about cool temperatures or cold temperatures or even freezing temperatures as killing microorganisms. It's more of just making them kind of go dormant. And then the final, like, real physical way that we talk about controlling microbial growth is through radiation. We have to be really careful with radiation because radiation isn't necessarily a thing that targets microbes, it's a thing that kills cells. And we are made of cells. So radiation can kill us too. Just like dry heat can burn you, too cold of temperature, like deep freezes, can hurt your cells. Right? So all of those things that hurt microbes can hurt you, so you have to be very careful. So if we look at our light spectrum, and this is why we make you take physics, it's part of the reason we make you take physics, right? You've got the visible light spectrum, and then you have these shorter and shorter waves. So you have ultraviolet light, like shorter wavelengths than the shortest visible light, ultraviolet. Ultraviolet is what we call non-ionizing radiation. Basically what that means is that exposure to UV light does not create like free ions or free radicals. What UV light does to all cells, not just microbes, is it creates what are called thymine dimers. And so what that means is if you have DNA that's like, right, this is one strand of DNA, here's the opposite strand of DNA, you would expect, right, this classic base pairing. That's what you expect. But what happens with exposure to UV light is these two thymines decide, well, they don't decide, they just do because that's what happens. They covalently bond to each other and they stop binding across the way, which is fine until the cell tries to replicate. And so when the cell tries to replicate, this strand replicates fine because it's normal. The DNA polymerase has no problem. This strand does not. Sometimes it won't replicate at all because DNA polymerase is like, I don't know what this means. Sometimes DNA polymerase sees these two T's that like in space are now very close together and it puts a single A. So it causes a deletion. But either way, you know, we haven't created any free radicals, we just created a problem here. So it interferes with DNA replication. It causes mutations. And eventually it can cause cell death. And when you're a single-celled organism and you die, not so bad. When you're a multicellular organism and your cells start to die, you get things like sunburn, which hurts really bad. And then your skin peels off. Constant exposure over time, you get cancer because of the constant accumulation of mutations in your cell over and over and over. And that's why people who have spent a long time in the sun are more prone to skin cancer, especially as you age. We typically don't see people your age who get skin cancer, though it does happen. But, you know, talk to your parents or grandparents or their friends, and somebody's always getting some piece of thing cut out of their face. Oh, I got another spot removed. That's my dad. Got another spot removed. I'm like, Dad. My dad was a landscaper for like 30 years. What do you expect? My mom, I'm just really lucky because all I did was sit out and tan, but she's got, you know, sunspots now. She's like, oh, my skin. I'm like, well, I bet it looked good, you know, when you were a teenager and you had a nice tan, but now, so that's what it does. That's that accumulation of mutations. But again, if you're a single-celled organism, you die. But if you catch it just right, you can do experiments with microbes where you expose them to UV light just for a little bit, and they can acquire mutations that make them resistant to antibiotics. It doesn't matter where the mutation is, it's just a mutation. So sometimes they'll get a mutation that allows them to be antibiotic resistant. So, you know, here you are, like here's tanning, is kind of what you're exposed to here. Now, what's more dangerous is ionizing radiation. These are the even shorter wavelengths of light, things like X-rays, gamma rays, electron beams. These create free radicals, a lot of like reactive oxygen species, etc. And when you have that build up in the cell, often what it does is it causes double-stranded breaks in the DNA. So like the whole chromosome just breaks. And our bodies are already pretty bad at repairing DNA. Like there's an enzyme that's supposed to fix thymine dimers, and we lost it during our evolutionary history. I'm like, we're naked, right? We don't have any hair even. And some of us are very pale, so we have no pigmentation to protect us. And we lost the one enzyme that's supposed to say, oh, no, do the right thing. So evolution doesn't always do the right thing. That's what it did. We lost the enzyme that can fix this. There really aren't enzymes that can take a broken chromosome and put it back together correctly. Often what will happen in the body or in the cell, in our cells specifically, is our body will recognize that we have the double-stranded break. We have this this repair system called non-homologous end joining. So let's say you have breaks in multiple chromosomes. The system is like, well, here's a broken end, fixed. But it could have put like chromosome 1 and chromosome 7 together. Here's a broken, fixed. Or maybe it does put the right one back together, but you lost some because it wasn't like an even break, so it'll kind of chew it up so it can put it back together. But a similar thing in terms of breaking the DNA is happening in bacteria. And that ionizing radiation, those free radicals, can also damage the way proteins are put together. So it's kind of a 1-2 punch of you've affected the DNA, but then you've also affected the proteins that exist that could have perhaps tried to fix the DNA. So you take out all of the machinery to repair, and that's why you get it to be bactericidal. So we can use ionizing radiation because we've talked about, okay, for sterilizing hospital equipment, right? Autoclaving, things that aren't sensitive to heat. Filtering heat-sensitive liquids. But what about like plastic things, like tubing, catheters, artificial joints? You want to put a catheter in the autoclave? What do you think is going to happen? It's going to melt. And then you have a pile of goo that you can't put in a person. You can irradiate it to sterilize it so that it's safe to put in a patient. And when we talk about food microbiology, we'll talk about foods that are irradiated before we consume them to keep them sterile. So microwaves are also on this light spectrum, and microwaves also have a microwave can have antimicrobial properties, but it's not because of the wavelengths of light, it's because it's hot. Right? So these wavelengths aren't going to hurt. It's just because it's hot. That's what the microwave does. Okay, questions about physical methods before we start talking about chemical methods. Have you guys gotten to the Kirby-Bauer assays yet in lab? Okay, well the faces tell me no. Okay, what are you doing in lab today? Morning lab. What did you guys do?
We just did we were doing growth.
Oh, you're doing the growth?
Yeah.
Oh, yeah. The serial dilutions? Oh, it's fun, right? Sorry, afternoon folk. You're gonna be here till like 5. It's a long one. I know. Yeah. I always tell myself, like, ask for the lab schedule so you don't have to ask the students, but I'm like, no, the students will tell me. They'll tell me what's going on. Okay. Well, so pretty soon you'll get to, because you're doing microbial growth, you'll get to control of microbial growth, and you'll use chemicals to see how they affect microbes. You'll do the physical methods too, but you'll get to the chemical methods. So again, one of the ideas here is that we use chemicals not often for sterilization, but definitely for disinfection. We want to reduce the number of microbes down as low as we can get it to make whatever it is as safe as possible. So how do we determine, though, if a chemical is good at reducing the number of microbes? So we can do various kinds of tests to figure out if a chemical is effective, what types of organisms is it effective against, and then of course we can talk about the different classes of agents that can kill microbes. So this is called a use dilution test. So you take a metal, or you can even sometimes take a paper, but usually this one is metal. You take a metal ring, you dip it in your test bacteria, and you place that in a disinfectant for 10 minutes at room temperature. Then you take the ring after it's been in the disinfectant and put it in culture media to see if the bacteria will grow. So that will tell you at 10 minutes, does this concentration of disinfectant kill the microbes? Even if there's one left, it'll grow. And again, this is one of those, well, 10 minutes, like, that's why they want you to spray your chemical or whatever on your countertop and leave it before you wipe it away, because it can take a while to reduce the amount of microbial growth. So this is called the use dilution test. What you guys will end up doing in the lab is a disk-diffusion. They're also sometimes called Kirby-Bauer assays, because Kirby and Bauer were the scientists who came up with it. So at the end of the day, if you remember Kirby-Bauer or you remember disk-diffusion, doesn't matter to me, they mean the same thing. To me, they mean the same thing. So whatever term you like. So this also evaluates the efficacy of chemical agents. What you do in this case is you have a disc, a paper disc, that has a chemical, or sometimes an antibiotic, which is a type of chemical. You make a lawn of bacteria, whatever bacteria you're going to test, and then you put that disc on top of where you spread the lawn. You let it grow, and what you're looking for is this area around the disc that shows no microbial growth. This does not mean that the agent killed the microbes. All it means is that it inhibited their growth. There could be microbes under there that you plated that can't grow. Doesn't mean they're dead. So what we call it is a zone of inhibition. And you often want to test a chemical like this or an antibiotic against multiple kinds of bacteria, because every chemical is not going to work the same against every microorganism. So in this example picture here, we've got staph, which represents our gram-positive. We've got 2 gram-negatives, E. coli and Pseudomonas. So they're both gram-negative, they're both rods. E. coli is in a group called the Enterobacteriaceae, which represent a lot of your GI pathogens. Pseudomonas is in a group called the Pseudomonads, or Pseudomonas-like bacteria. This one tends to be very good at contaminating and causing nasty infections. And it's even though it is gram-negative, like E. coli, biochemically it's very different. So it's good to test multiple. So what you can see is, let's just look at chlorine. Chlorine was pretty effective against Staph aureus. It inhibited the growth. We have a nice big zone of inhibition. Because what's happening when you put these discs on the plate, the chemical diffuses out. That's why it's a disk-diffusion assay. The chemical diffuses out into the growth media. So you would have the highest concentrations right near the disc, lower and lower concentrations the farther away from the disc you go. So what it's saying is that these edges, even when you had a pretty low concentration of chlorine, you still inhibited the growth of staph. When you go to E. coli, I don't know what happened up here, but down here, looking at it, maybe the zone isn't quite as big. When you go to Pseudomonas, the zone is even smaller. So that tells me that at that concentration, whatever concentration you put on the disc, Pseudomonas doesn't do too bad. which is a bummer because chlorine is what you use to put in your pools to control microbial growth. Pseudomonas is what causes swimmer's ear. I know, right? And then, I mean, just for funsies, look at the other three, right? Everybody worked a little bit against staph. The phenylphenol, and this is a compound called quat, it's like an ammonium-based compound, worked-ish against E. coli. This hexachlorophene did not, so you wouldn't want to use that against gram-negatives. And then nothing else worked against Pseudomonas. Pseudomonas was resistant to all of those other chemicals, like completely resistant at those concentrations. I am almost certain that as long as you do the Kirby-Bauer assay correctly with your antibiotic discs, you'll find that Pseudomonas is also resistant to most antibiotics. Okay, it's a nasty one, but it allows us to determine. This is really common in a clinical lab setting to do this with antibiotics. Okay, because there's a standard, there's an American standard and there's a European standard. Where it'll tell you each species, it'll tell you each antibiotic and what diameter of zone of inhibition you're looking for. If the diameter is, bigger than the cutoff, the bacteria is sensitive, you're good to proceed with that antibiotic. If the diameter is smaller than the cutoff, the bacterium is considered resistant and you should not use that antibiotic for that patient. It's literally like one of the biggest Excel sheets I've ever seen because it's like all of the antibiotics and all of the bacteria. So this is really common, those of you going into clinical medicine. So we want to use chemicals because we can use them to disinfect or as antiseptics. Right? Remember, disinfection is what we would do to a non-living surface, an inanimate object. Antisepsis is what we do to a person, so we don't disinfect a person. Even though colloquially we say that, but technically antisepsis is what you talk about with a person. And just like you would expect, these chemicals that act as disinfectants or antiseptics interfere with the major targets in the cell membrane, the DNA, the proteins. If you can interfere with how the microbe functions, you interfere with it being able to survive. Okay, so on the next few slides, I'm calling it like the parade of chemicals. You don't have to know all the chemicals, okay? I'm one of those like, yeah, this one you could look up. You don't have to know all of the things. So I'm gonna do the parade of chemicals very quickly and understand that I'm not gonna ask you about all the chemicals and what specifically they do, just that they exist and they do things. So phenols are really good at killing vegetative cells. They cause membrane damage. This is like Rocol is a phenol, Lysol is a phenol. They damage the membrane. And these are really common as disinfectants. You can also use them as antiseptics at pretty low concentrations. They're a local anesthetic. So when you take not a cough drop to suppress the cough, but if you have a sore throat and you take a lozenge that kind of like reduces some of the pain, it's usually a phenol compound. And they can be antibacterial at higher concentrations. At even higher concentrations, that's when you would use them as a disinfectant, like Lysol. Do not spray Lysol in your throat. Okay, I feel like that's a disclaimer I actually have to make. Higher concentrations are not good on people. Lower concentrations that you can buy at the pharmacy, please feel free. Okay, so again, damage to the membrane. These are phenol derivatives. Again, they disrupt the membrane. There was one that was really, really common called Triclosan. It was found in all of these kinds of products. And I know you might not be able to see all of them, but you have everything from like Ajax, deodorant, dish soap, Clearasil, toothpaste, makeups. They all had this triclosan. It turns out triclosan makes it through the water treatment plants and gets into the water supply and kills all the microbes in the water. And you don't want to do that because you need microbes in the water. So we're not allowed to use it anymore. But again, these are phenol derivatives. They disrupt the membrane. We can also use things like halogens. Iodine is pretty common, especially in a clinical setting. I'm sure you've heard of betadine, right, or iodine. It impairs both protein synthesis through kind of creating reactive oxygen species. It also alters membranes. Chlorine is another halogen, which we just talked about in the context of in pools. Okay, but I don't know if you knew this or not, but bleach, the really major chemical component of bleach is chlorine. It's hypochlorous acid. This is, again, used for disinfection of things like drinking water, dairy equipment, food industry surfaces. You probably have it at home. Maybe you put it in your laundry to brighten your whites, or maybe you dilute it and use it as a cleaning product. Either way. Chlorine plus ammonia is what we use to disinfect drinking water. So eventually we try to remove the chlorine from the water before it goes back into the water supply, but we add chlorine to the water to disinfect it, get rid of all the bacteria in there. Alcohols, again, another very common, right? Every time I'm like, ooh, alcohols, right? Your hand sanitizers. They denature proteins. They can also dissolve lipids. But one of the things you have to consider is you don't use 100% ethanol. Like, it might sound like, well, I want the strongest ethanol possible, right? I want to get rid of as much as possible. But when you have a really high concentration, what happens is it kind of dehydrates the cell a little bit, but not enough to kill it, but enough to prevent the alcohol from penetrating into the cell. The membrane kind of locks up and doesn't let the alcohol in, so it has no effect. So what they're showing you in this slide here, in the table, the highest concentration of ethanol, and they exposed Streptococcus pyogenes. This is the bacteria that causes strep throat. They exposed it to 100% ethanol for 10, 20, 30, 40, 50 seconds, and the bacteria grew. So it didn't kill them. But as soon as they added just a smidge of water, 95% alcohol, 5% water, no growth, no growth, no growth, no growth. Because that little bit of water allowed the alcohol to penetrate in where it could denature the proteins. You can, of course, dilute it too much. So 50% is effective, but only for longer exposures. And 40% is no longer effective. It's just too much water, not enough alcohol. So usually these will be between 60 and 80%. This one is 70. That's the most common I've seen. Yeah, this one is also 70. 70% ethanol. So after 10 seconds, it would kill the bacteria that causes strep throat. Maybe even faster, but that's what they tested in this experiment. So again, if you've ever, you go to CVS and you get your flu shot, please get your flu shot. The flu is terrible this year. Right? They take a little alcohol swab to make sure there's no contamination, and then, or if you take an injectable medicine, I'm sure you have to, you use an alcohol swab to clean the injection site. If you're diabetic and you have to do your finger prick every day, right? Anytime you have to make an ouchie on yourself, you should use alcohol. Not after, that hurts. The alcohol after burns. We can also use heavy metals, things like silver, mercury, copper, and zinc. These typically will kill cells, and they do it by interfering with the sulfur groups in amino acids like cysteine and methionine. Okay. So really commonly in tertiary and even quaternary structure of proteins, you have those cysteine and methionines that can form covalent bonds. They call it a disulfide bridge. These metals will interact and break that bridge apart, which causes the three-dimensional structure to fall apart, which causes the protein to no longer work. And, we joke in microbiology about, hey, they used to just give people mercury and then they went crazy because that's what happens. Or they would give people arsenic and then they went crazy. But, these actually are still really commonly used, of course, not just like straight up injected into your body, but, right, things like silver nitrate can prevent eye infections in newborns. Silver compounds are really, really common Also to put on patients who have a burn wound. You'll put a silver-based ointment over the burn. Paint, especially paint that goes in like bathrooms and kitchens where there might be a lot of moisture, has mercury chloride in it to prevent mildew formation. If you have any of these things, a tank, a pond, a swimming pool, like aquatic plants, you can use copper sulfate. There's a copper containing hand sanitizer, zinc. This one is really common in anti-dandruff shampoo, because really what dandruff is it's usually just like a little bit of an overgrowth of a fungal infection. So if you ever have dandruff and you're like, what shampoo should I get? Look for one that has zinc. Clear you right up. If it doesn't, go to the doctor. They'll give you a prescription strength one. It'll clear you right up. Okay, so if you buy Head and Shoulders and you're like, why is it not working? Make sure it has zinc. Okay. Zinc chloride found in mouthwash because you don't want stinky bacteria in your mouth. All of these metal-based compounds, they're even starting to make hospital beds that have copper handrails because it's antimicrobial. So that when people get in and out of the bed, right, there's not microbial contamination. So using heavy metals has a really long history in treating infection, even before we knew what caused infection. But of course, now that we know and we can control levels, right, we still use these heavy metals because they have really great biocidal action. We also use soap, hopefully. We have this conversation almost every day. I mean, when you teach microbiology, you talk a lot about soap. Because it helps to emulsify, like it breaks them up into like more particles that you can kind of like wash off. It doesn't necessarily kill microbes, but it helps to solubilize them so you can physically remove them from your hands or your face or whatever on your body. We have other surface active agents as well. Ammonium-based compounds are bactericidal. What these do is they act more again like a detergent. They disrupt the membrane. So they're not necessarily getting into the cell right away. They want to disrupt the plasma membrane, and then they can get in and start to denature the proteins. So a really common one is benzalkonium chloride. So it's found in mouthwash, hand soap, various, like the alcohol-free hand sanitizers usually have benzalkonium chloride. Now this one doesn't because it's a pure alcohol hand sanitizer. But some people don't like the alcohol-based hand sanitizers. Benzalkonium chloride is very good. And some of these other compounds that are not toxic to humans, there's a lot of research that goes into using them in conjunction with antibiotics to help combat antibiotic resistance, because there's a couple of different ways you can target the microbe. And again, I'm not going to ask you about all of these. This is the parade of antimicrobial compounds. Aldehydes also work really well. You'll notice they inactivate proteins. They also can interfere with DNA because of where they bind. They bind ammonia groups, they bind hydroxyl groups, they bind carboxyl groups, they bind sulfhydryl groups. Those are basically all the functional groups of amino acids in DNA. Formaldehyde is a good one that we typically don't use anymore because it's terrible. If you've ever dissected something that smelled really, really chemically, may have been preserved in formaldehyde. We try not to use that anymore because it's gross. Glutaraldehyde is actually really effective. This can also be used as a liquid to disinfect hospital instruments. A 2% glutaraldehyde solution is actually the most common liquid sterilant. Like if you wanted to dip something in a liquid sterilant, glutaraldehyde is really good. And again, because these are great for inhibiting microbial growth, they are also used for embalming. I mean, that's the whole point, right? You want to control microbial growth so that you can have, a funeral service or something, and not have to experience what actually is happening. So formalin and glutaraldehyde are really, really good. We can also use gases as sterilants. Again, they work with nucleic acids and proteins. They're very good at being used for sterilizing heat-sensitive materials. So you can use some of these gases if you don't want to use like a liquid like glutaraldehyde. You can use things like ethylene oxide, like a mattress. Yes.
I had a question for the other slide.
Sure.
Instead of formaldehyde,
Mhm.
like cadaver rooms, or like cadaver labs, what else would they use instead of?
I don't remember what they use. They may have transitioned over to more like glutaraldehyde because it's less pungent. There's nothing wrong with formaldehyde. It just really stinks. Yeah, it made one of the girls in my biology class in my freshman year of high school, she just like passed the heck out. And we were like, are you afraid of fetal pigs? Because we were doing a fetal pig dissection. She was like, no, it's not the pig, it was like the fumes. She just, whoosh. So anything that produces less fumes. But gases are, again, also really common. You all are probably far too young to remember, but immediately following the September 11th, there was, it was like in October of 2001, there were letters mailed across the eastern seaboard that had anthrax in them. And they were mailed to places like senators and representatives in the halls of Congress. And they were literal anthrax spores. People died. They were transiting through like the postal system, in newsrooms of these various companies. How do you autoclave like congressional buildings? You don't, right? And you can imagine that all of those Congress people were like, I'm not going back to work when there's anthrax spores in my office. Fair. You use gases. You pump that room full of gases to sterilize the spores. That's what they did. So these gases, yes, have use on things like mattresses and plastic syringes and medical equipment. You can irradiate some of those same things. But the gases are really good. And so that's how you could sterilize, if you had to, a building. You can also use chlorine dioxide, right? So here's the one that they used for the anthrax endospores. These gases are really good. Okay. So to fumigate buildings. It's not a situation we typically come across where you have to fumigate an entire building from anthrax spores, but it's not unprecedented. So there is a way to do it. Other forms of oxygen can be used. So anything that causes reactive oxygen species. So we use things like hydrogen peroxide as an antiseptic. It can sometimes also be used as a disinfectant. Of course, we know that some microbes are capable of dealing with hydrogen peroxide because they produce an enzyme called catalase, so it doesn't have any effect on them. Peracetic acid, ozone, benzoyl peroxide, anything with peroxide. Benzoyl peroxide, maybe you have something at home that has that. It's often used for acne in your acne medication. So if you have like a, face wash that you use, check the label, maybe it has benzoyl peroxide. But it's really good against anaerobes, and anaerobic bacteria are the ones that cause acne because they get really deep in the pore, and that's what causes the infection. So you got to get rid of the anaerobes. Okay, so that's the parade of antimicrobial compounds. And again, I'm not going to expect you to remember all of those. But what is important is what are the common mechanisms of action? Right? Membranes, DNA, and proteins. And under what situation might you need to use one class? Maybe heat versus liquid versus filtration, right? Radiation, gas, like in what situation might you need to use one type of chemical or another? But I'm not going to expect you to remember what phenols do. Okay. We also want to preserve perishable products because things are expensive and we don't want them to go to waste. And so we know that there are lots of food products that will spoil rapidly, especially our fresh foods. right? Fresh meat, fresh vegetables and fruits, bread with no preservatives, it will all go bad pretty quickly. And so the goal is to prevent spoilage so that we can increase shelf life. Again, you're going to be really ticked off if you spend like $4 on a thing of strawberries and you open them the next day and they're rotten and you can't eat them. So, what are ways that we can preserve foods in order to keep them shelf stable so that we can eat and not waste our money? Well, we can add chemical preservatives. Sulfur dioxide is added to wine, organic acids to control molds in things like bread, cosmetics, and other acidic foods. Right? And so you can see here in this bread from Trader Joe's, ascorbic acid. It's a preservative. It's to help prevent fungal growth. Right? Fungi are what make bread go bad. You got that nice little blue fuzzy thing. Oh man. Do I rip it off and still eat the bread? No, I'm not going to do it. Okay. Nitrites and nitrates. They can prevent botulinum spores from germinating. And I don't know how much you call them meat, but let's call them, let's go with meat, that's fine. Preserves the color of meat like ham, sausage, bacon, and hot dogs. Maybe that's in order of the amount of meat that's actually in them, I don't know. That's why they're all kind of the same color, kind of pinky color. Okay, and then you can add antibiotics, especially to inhibit endospore forming bacteria and to inhibit bad fungi, for example, in cheeses. These are a little bit different antibiotics. They're not like the antibiotics you buy over the counter. They're called bacteriocins, or they can be like active against fungi, and they usually just punch holes in the cell. Of course, really commonly, and we don't even really think about it, but we use low temperature. Okay. I love that, like the Trader Joe's sign, I didn't come up with these pictures, but the Trader Joe's sign that's like, our breads have no preservatives, but the previous one was the bread with ascorbic acid, which is technically a preservative. So I don't know what Trader Joe's uses as their definition of preservatives. I like TJ's, but it's all good. It, again, stops the growth, especially of those mesophiles that grow at room temperature or warmer. But some of the ones that prefer cooler temperatures will still grow, and some of the ones like Listeria that just don't care will still grow. Freezing stops microbial growth. Some of the cells may be killed, but most of them not. Most of them will still be viable. And then, of course, we can also, like when we talked about hypo and hypertonic solutions, we can reduce the water availability. So, you know, classically, I think the example that somebody mentioned in class was salt. Yeah, absolutely. Salt is great. That's why salt used to be like more, you know, more valuable than anything. More valuable than gold and diamonds is salt, because you can preserve your food. Sugar does it as well. So that would be like up there with your jams and jellies. I mean, like salty jam. I like sweets. I don't think I would like salty jam. So we'll use sugar instead for the jam. But you know, beef jerky also probably shouldn't taste sweet, so salty for there. Drying will help because that removes the water, and then you can supplement it with salt or sugar to prevent any other microbes that could have survived the process from growing. And then, of course, freeze drying can be pretty common. Coffee, milk, meats, fruits, vegetables. So drying stops microbial growth but doesn't necessarily reliably kill, which is why you might supplement with something like salting or adding sugar. So like those dried fruits will usually have sugar added. Your dried meats will have salt added as kind of just an extra prevention of microbial growth. But later in the semester, we'll talk more about food microbiology and how we can use microbes to, or how we can control microbial growth in food, as well as how we use microbes to make food. Okay, questions about control of microbial growth before we move on a little bit to antibiotics? Yes.
So, like for this food preservation, like bread and stuff.
Uh-huh.
For people who like see it and say, oh, it's all these acids and stuff like that, and complaining? Are they is it like, are they complaining for no reason, or is it actually bad for us?
Yeah, they're just complaining for no reason. Yeah. I mean, people get all feisty about GMOs too, right? Like, oh, GMOs. Like, 98% of the corn in the United States is GMO. Like, good luck finding non-GMO corn. You know, GMO and like those types of things added for preservation are why you have like low-cost products. Like, that's why things like bread, well, I mean, not anymore, but it used to be cheap, right? You can get a loaf of bread at Walmart for like $1 because it lasts so long. If bread was going bad all the time, it would be really expensive because it would be expensive for them to produce and ship and put on the shelf, and then it would get rotten. So the prices would go up the more, like the less freshness, I guess, right? The more preservatives you add, the longer it lasts, right? Same with pasteurizing milk. Raw milk won't last very long on the shelves. If you don't refrigerate it, how long do you think milk is gonna last on the shelf? It wouldn't even last from the dairy, right, to the store. So adding preservatives makes those food products more accessible to more people, and they're not known to have any harmful effect. Yeah, I mean, it's the same thing with the GMOs where they make plants that are like drought resistant. If a farmer loses their whole crop to drought, that's what happens to the price of that crop. But if the crops are drought resistant, it helps save money and keep prices lower. And again, there's no known harm from them. At the end of the day, everything causes cancer, unfortunately. It's just kind of, I mean, California has a law, you have to like have that sign, Prop 65, like Starbucks, like coffee, roasting coffee causes cancer. The roasting process makes carcinogens. I mean, I still drink coffee every day. Okay, so I jumped from 7 to 20 because 7 is the control of microbial growth. 20 is antimicrobial compounds. So I feel like it kind of flows together. Not how they organize the book, but that's fine. I can do what I want. So I put 20 after 7. So we've already talked about the idea that the poor Dutch tailor discovered microbes and nobody really cared. And then eventually people started to care. And then in chapter 1, we talked about this idea that Robert Koch was able to link that specific microbes can cause a specific disease. We call that the germ theory. We'll talk more about Koch later. But now the question is, how do we treat diseases? So in the previous chapter, we talked about how can we control microbial growth as much as possible to prevent infection. But we also know that infection happens. So how do we get rid of infection when it happens? So the goal was to search for what we called the magic bullet, a compound that would target the microbe and not hurt the host. Because yeah, mercury will kill microbes. It will also kill you if given enough. So we want to make sure that we have something that will kill the microbes and not harm the host. So an antimicrobial drug is a chemical that interferes with the growth of a microbe in a host. Antibiotics are substances that are produced by microbes, generally, to inhibit the growth of another microbe. And chemotherapy is the use of drugs to treat disease. And again, broadly and colloquially, when we talk about chemotherapy, we typically are talking about anti-cancer treatment, but chemotherapy literally just means chemicals to treat disease. Infection is disease. So treating infection with chemicals is chemotherapy. And so the history of chemotherapy, we often start here with Alexander Fleming. We'll go back in time a little bit, but that's okay. He discovered penicillin because he had bad lab habits and he left a bunch of plates on the lab bench when he went on vacation. And when he came back, he saw that in some plates there had been fungal growth, but the microbes couldn't grow where the fungus had grown. He saw like zones of inhibition basically around the fungi. So they called it mold juice, which is not a very attractive name for a compound. Yeah, well, come get your mold juice today. They ended up purifying the actual compound in the mold juice that had the antimicrobial effect, and that was what they called penicillin, because the mold was in the genus Penicillium. So the first clinical trials were in the 1940s, and it was mass produced during World War II for the treatment of soldiers injured in combat, and also any war workers who might have gotten sick. It was to keep people working for the war effort, whether they were at home or abroad. What we now know is that there are huge numbers of microorganisms that produce compounds that can kill other microorganisms. Antibiotics exist as microbial warfare. You are in my spot. I want to be in that spot, so I am going to kill you. And when you're all single-celled organisms, there's no teeth and claws. I can put out a chemical that kills you. So we get a lot of our chemicals that kill other microbes from microbes. And actually, the majority of them come from this group right here, Streptomyces. These are all soil bacteria. Really, really common from soil bacteria. Some of our antibiotics do come from fungi. Some come from other gram-positive bacteria, but a lot come from the soil. Amphotericin B, this allows them to compete with fungi. Amphotericin B is a really good antifungal. But chloramphenicol, tetracycline, erythromycin, neomycin, streptomycin, gentamicin, those are all antibiotics. Maybe you've even been prescribed them. So they're antimicrobial warfare. Now, the majority of antibiotics that we have are either semi-synthetic or completely synthetic. So by semi-synthetic, what I mean is that there was a naturally occurring compound that was isolated from a microbe that had antimicrobial activity, but in some way it has been chemically modified in the lab. Maybe to combat resistance, maybe to improve the spectrum of activity to allow it to work against multiple kinds of bacteria, maybe to allow it to make it through the GI tract so it can be given orally. But there are lots of reasons scientists might modify a naturally occurring antibiotic to improve it in some way. So we call those semi-synthetic if they've been improved. And then synthetic antibiotics are made completely in the lab. So this is where especially like computational chemistry comes into play, because you can do a three-dimensional model of a protein that you might want to inhibit or a target that you want to inhibit, and you can computationally design compounds that should interfere with like that enzyme or that protein, and you can synthesize them in a lab. And when they do that, they make libraries of literally like tens of thousands of compounds that could potentially have antimicrobial activity. So one of my friends, for his PhD project, he tested tens of thousands of compounds against Candida albicans, which is a fungus, so it was antifungals, but same idea. And very few of them actually work. So it's just that broad screening to see when you actually get an effect. So synthetic antibiotics are also very good. It just requires a lot of testing. With a semi-synthetic, we at least know the base is going to work because the natural compound works. We just want to make it better. So when we talk about antibiotics, they'll usually have one of two functions. They either will kill the bacteria, which in some ways, maybe that's what we want, or maybe not. If you have a patient that has a really severe infection with gram-negative bacteria and you start killing all of them, you release all of that LPS, all of that endotoxin, and you cause them to go into shock and they can die. So maybe you don't want to explode all the cells. Sometimes, though, it's good enough if you can just prevent them from growing. That can stop the course of the infection. That can allow the immune system to come in and start clearing things up. But, you know, when you have patients who are immune compromised, for example, you gotta go for the big guns because they don't have a way to get rid of it otherwise. And that phenomenon of being the magic bullet, the scientific way we talk about it, is selective toxicity. So selective toxicity means it will inhibit the growth of or kill a microbe without damaging the host. So that's selective toxicity. Broadly, we achieve selective toxicity by targeting features of a microbe that are not found in people. So what would be found in a microbe that's not found in people? Cell wall. That's a good one. What else do microbes have that's different from people? (70S) ribosomes versus the (80S). Microbes also use slightly different proteins to replicate their DNA and to do transcription. Microbes have slightly different cell membrane components. We talked about one other mechanism in chapter 5 when we were talking about enzyme function and enzyme inhibition. And we talked about a class of antibiotics that works really well against microbes but doesn't affect people. This is where the chapters all come together. Not statins, but a similar idea where you inhibit an enzyme, but this one's an enzyme that only bacteria have because they need to make something that people can't make.
Oh, is it the purple one?
Yeah, folic acid biosynthesis, metabolic pathways. So you inhibit a metabolic pathway that a microbe doesn't have, or that a microbe does have that we don't. Microbes have to have folic acid, so do we. But we eat it, we import it. They have to make it. So we stop them from making it. They're not capable of importing it, they die. So our major targets for antibiotics are definitely the cell wall, the ribosomes, protein synthesis because we do it differently, DNA and RNA synthesis because in some ways we do them differently, the cell membrane because there are some differences, and metabolic pathways that they have that we don't. Those are going to be your 5 major targets for antimicrobial therapy. So we'll pick up with antibiotics on Tuesday. Have a good weekend.